Bismillahirrahmanirrahim. Assalamu alaikum friend. Today we will start a lecture on pyrosequencing. So pyrosequencing means to remove pyrophosphate from the addition of nucleotide. So basically this sequencing is uh, depend on the sequencing by scientists. And the principle of this sequencing is sequencing by scientists. So, when a new nucleotide are added to the unknown template strand of DNA, so the pyrophosphate are removed. So, as that sequencing is called pyrosequencing. So, let's go to explain that how the pyrophosphate are removed from this sequencing. Now, this is the structure of nucleotide and sorry. In carbon number first nitrogenous base and here the OH H and here the OH and phosphoric acid are present in carbon number 5. And then this is the another nucleotide. We explained that how pyro phosphate are formed during the sequencing of pyro. So here the this is the another DNTPs. So when this new nucleotide are come here to attach with the the previous nucleotide of DNA. So in this 5 carbon, this phosphate are removed, and here the hydrogen are removed, and water are eliminated from this molecule, and oxygen bond make bond with phosphate. So here the make bond with the carbon. So here the two phosphate are removed. So these phosphate are called pyrophosphate. When basically this all process are depend upon in this pyrophosphate. When pyrophosphate are removed, so in pyrogram we show that the nucleotide are added, and light are produced. So when this phosphate pyrophosphate are formed, so they this pyrophosphate are combined with another type of substrate molecule which is called APS, and this APS is stand for adenosine 5-phosphosulfate. So when this pyrophosphate are formed, this pyrophosphate are combined with APS substrate in the presence of enzyme called ATP sulfurylase. So the function of ATP sulfurylase to form ATP molecule from the pyrophosphate and the substrate APS. So these two are combined and they form ATP molecule. During the sequencing, this ATP are required to convert another type of substrate molecule which is called leuciprin. In this ATP molecule, this substrate molecule are converted into oxyleuciprin. and the presence of ATP molecule. When this leucocyprin are converted into oxalocyprin, so here the light are produced. So this is the mechanism of the pyrophosphate production, and how this pyrophosphate help in the pyro sequencing. So now I want to repeat this process. So here, when during the sequencing, when new nucleotide are added to previous nucleotide and to unknown sequence, so pyrophosphate are removed, and then this pyrophosphate are combined with APS, adenosine pyrophosphate, and then in the presence of ATP sulfurylase, this type of um, substrate molecule and pyrophosphate molecule are converted into ATP. They form ATP molecule, and this ATP molecule are helped to convert leucocyprin into oxalocyprin, and light are produced. So this light are very important for the pyrosequencing. Now, how we can sequence a unknown template strain of DNA molecule. So let's go to start the sequencing of an unknown template DNA molecule. Suppose this is the unknown template DNA molecule strain. And first step, we attach the pipe strand site with the substrate molecule called adapter, which already which we known already this sequence. And here we attach uh, primer and design, and this side we add primer. So, and second step and first step, we can only do adapter linkage with the template strand, and uh, attach to this adapter this known sequence primer. And the second step, we add nucleotide. These nucleotide are basically DNTPs, and these DNTPs are present in four type of tubes, which is basically D, A, T, P, S. Alpha S. This type of ATP are modified because to not use this ATP to convert leucocyprin to oxalocyprin. Is that why this ATP are this adenosine triphosphate nucleotide are uh, modified, and then the second tube DCTPs present, and the third tube DTTP and GDGTP are present. So remember, we add only one type of at a time, one type of DNTPs only. That may be the DATP, there may be the DCTP, and there may be the DTTP, and also. So when we add this type of DATPs, so when uh, the complementary strand, the complementary nucleotide are present here, so attached they attach DA adenosine are attached. So here, when this adenosine are attached, so there are previous nucleotide are present. So pyrophosphate are pumped. This pyrophosphate are converted into this pyrophosphate combined with APS, and then this APS make ATP synthesis, and then this ATP used leucilprin and converted into oxyleuciprin. So here light are produced in the pyrogram. We show that the light are produced, so we indicated that the nucleotide are attached. So here the complementary are present. So when A are attached, so there are an unknown triplet, the T are present. When we add DCTPs and they are attached, so here light are produced, and this who are all process are cycling palm. So when we add DCTP, so here in the complementary G are present that are show in the pyrogram. So if we add DTTP and they are not attached, so here pyrophosphate are not pumped, then the leuciprin are not converted into oxalociprin, so light are ATP are not produced, then if the ATP are not produced, so here the light are not produced, then we in pyrogram we have not show the peak of D TTP. So we add another type of enzyme which is called apyrase. This type of enzyme is helped to remove the unmatched nucleotide. So it's remove this type of unmatched nucleotide from this sequence. So here this is the all about pyrosequencing. If you learn something from this video, so please like, share and subscribe my YouTube channel.